Well, good afternoon, everyone. I'm Athol Johnston. I'm the analytical service. I'm the science director at Analytical Services International, and I'm here representing the Royal College of Pathologists (SAC). Thank you for joining today's webinar, and it's on analytical considerations for detecting and quantifying novel synthetic opioids, including the nitazenes in biological samples. Today's webinar will run for approximately 1 hour and will include a short presentation and there'll be a question and answer session at the end. So if you want to ask a question, use the Q&A function at the bottom of the screen. Please note the webinar will be recorded and attendees can self-credit 1 CPD point for attending today's webinar. As I said, the webinar comes from the SAC in toxicology, but today it's Lewis Couchman, who is currently the Facility and Research Director of Analytical Services based at St George's, University of London. He'll be presenting this. His current role involves overseeing the development, implementation, and routine analysis of biological samples for therapeutic drug monitoring, forensic and clinical toxicology, and support for clinical trials. Lewis completed his BSc degree at Loughborough University, then did an MSc and clinical science training at Queen Mary, University of London, and King's College Hospital. He's got a PhD from the University of Leicester, and additionally, he's a committee member of the Society of Chemistry Separation Science Group and the London Biological Mass Spectrometry Discussion Group, and he's a past chair of the London Toxicology Group. So over to you, Lewis, and let's hear the presentation, please.
Thank you very much, Athol. Sorry to give you so many acronyms and in that
I didn't get too many acronyms in
there. Thank you, everyone, for joining. I'm just sharing the screen, which hopefully you should now be able to see. You confirm that, Athol, then I'll carry on.
Yes, I can see it.
Perfect. Okay, so yeah, thank you very much. The title of today's talk is the sort of the subtitle here. Perhaps the more catchy title is this quotation, which I'll start with today. Crossing my fingers once more into the abyss. The reason I start with that is that this specifically relates to a synthetic opioid. Fortunately for us, a very rarely encountered synthetic opioid. It's a utopioid, U47700. But the quotation at the top of the slide there comes from this paper back in 2021, who interviewed some drug users, in particular referenced this website called flashback.org, which is translated through from Swedish, and it's a really useful resource for us, as are other sources of information that I'll come to over the coming slides. But really, if you want to take the sort of the main theme of today's presentation, it's how we in the laboratory setting are trying as best we can to keep up to date with the synthetic opioids that are beginning to find their way into the illicit drug market in the UK. Obviously, lots of this coming across from the United States, but certainly these compounds are in our drug supply in the UK, and we are seeing them in coronial cases. So this quotation, as I said, it comes from a particular drug user, and with reference to this U47700 drug, which, as we'll see later on with the nitazene opioids, has its roots as a pharmaceutical product. This one from Upjohn Pharmaceuticals in the 70s. And this compound specifically has a potency at the opioid receptors approximately 7.5 times that of morphine. And this sort of notion of relative potency is something that we'll come back to, and it's certainly something that's very useful for us in terms of sort of trying to keep on top of how we test for these compounds. Other sources of information for us, just in terms of knowing what people are taking and what sort of compounds we should be looking for in our biological testing, is just personal correspondence with medics and people involved in treating drug addicts. So Mike Kelleher, if anyone in the audience knows him well, is an addiction psychiatrist in London. And this is a quotation from one of his patients who's moved to the local area specifically looking for fentanyl, um, with the impression that the fentanyl that he gets in London lasts longer and is cheaper than the gear elsewhere. So some evidence here that people do travel to try and find what they think is a good dealer. How you measure good there, I'm not quite sure, but people do potentially travel to where they think they can get better drugs. And in this instance, particularly, it's of relevance because despite this user thinking he was getting good fentanyl in London, he wasn't getting fentanyl. It was in fact a nitazene opioid. The other thing that was interesting for me, this is going back now a year, 18 months or so. People are actively seeking these novel psychoactive substances, fentanyl analogues, and more recently the nitazenes. So experienced heroin users not just seeking more heroin, but almost actively seeking out these alternatives to the more traditional drugs of abuse. And also, there are people taking these things who do not die all the time. So we're not only seeing these in coronial cases. There are now plenty of examples of people taking these more recreationally and not overdosing and ending up as a post-mortem case. The other thing that's really useful for us in the lab to have a feel for what's going on in the drug market is to think about drug checking services. Now, there are a number of these home drug testing kits that are available online. There are some scientific reports of them formally testing those and reporting on the performance of some of these kits, which is not always good. So certainly they are sold as and perform as presumptive tests only. They don't necessarily indicate or account for any level of dose or purity, and they'll be geared towards specific classes of compound. These testing kits exist for both drug products themselves and also urine and oral fluids, so some biological testing. So they exist, and knowing what these drug testing kits are and what is available is a useful thing for us to know. But perhaps of more use are more formal drug checking services, and the one that we keep a close eye on is the Wedinos service, which operates in Cardiff. So it's a laboratory-based testing service. They use the same analytical instrumentation, more or less the same analytical instrumentation that we use in the laboratory to test the biological samples. But Wedinos, if you've not heard of it, is a service whereby drug users can send samples of their drugs through the post anonymously, and they get tested, and the results are then posted online via a unique reference number. The other thing that's useful for us here is the submissions of these samples. There is an optional record of what the purchase intent was, and so it's quite interesting then, and I'll give you some examples on the next couple of slides of what people think they're buying and what the drug content of those samples actually are. There's also a number of other drug testing services available. You may have heard of front of house or in-field testing. For example, at summer music festivals, there's an organisation called The Loop that do that. There are others which are more back of house testing. So there's a company you may know called Tic Tac, who we have the benefit of working very closely with here at St George's, who also do an in-field testing. So they'll take a mini lab, if you like, to a music festival, but they also have a laboratory-based confirmatory setup as well. So there are some limitations of the in-field testing, specifically when it comes to the more potent opioids that may be present as contaminants or low-level fillers in tablets that you can't do quite so in the field, and hence the requirement for a laboratory-based backup to that testing. Wedinos, their website publishes annual reports as well as monthly reports of things that are submitted and what they contain. Just a couple of snippets from recent reports that may be of interest. So the 2022-23 annual report from Wedinos suggested that almost 40% of the samples submitted when compared to the purchase intent did not contain what was expected, which in itself is an increase year on year from the report the previous year. Between, as you can see, April 22 and March 2023, specifically with relation to the nitazenes, there were 36 samples submitted that contained a nitazene, but none of those were listed as the purchase intent. Again, with specific regard, the nitazenes, 64% of 36 samples that contained nitazenes were purchased thinking that the product was oxycodone, and 22% the purchase intent was for the user to be buying a benzodiazepine, but these samples actually contained a nitazene. Two slightly more recent reports from the back end of 2024. So in August 2024, 12 samples. So just in that 1 month period, there were 12 nitazene containing samples with the compounds listed there. We'll come on to the specific nitazene compounds, which ones are controlled in the UK, etc., and some of the chemistry related to them in the next few slides. But the one of note there is the last one on the list, the azoto nitazepine, which is a newer nitazene analogue that currently is not controlled in the UK on the list of compounds most recently issued from the government. And in September 2024, just one nitazene containing sample from London, which was a green tablet that again contained this newer nitazene analogue that's not on the UK list of banned substances. Just to show where some of these nitazenes are being found, clearly there are a number of the samples that are purchased as heroin that contain the nitazene opioids. But a number, as I also mentioned, were purchased as benzodiazepines, some as diazepam. This I assume is street diazepam. Similarly, street Xanax or alprazolam, which contain two different nitazenes. Oxycodone, I mentioned. There are a number of samples that were purchased as oxycodone but contained one or more nitazene analog. The others that are more perhaps worrying, there are synthetic cannabinoids, so a completely different class of compounds but found to contain nitazene analogues. 2CB is an interesting one because that, of course, most commonly would be seen as a party drug, as a stimulant type drug, but the presence of nitazenes there would be very much, I would expect, not what the user would intend. And also the presence of the two of these nitazenes in THC containing vaping products. Which is interesting. More recently, there has been one submission to nitazene, to Wedinos, where the purchase intent was etonitazene, and that in fact contained etodesnitazene. So even an example there of someone thinking they were buying a particular nitazene but getting a different analog. So when it comes to the relative potencies of the different nitazenes, there may be some considerations there, and I'll come back to that a little bit later. There are drug user forums. One example is Erowid. This is just an example from the Erowid website. So this is where drug users will report their experiences on particular drugs. There's one, or there was certainly one when I last checked, back end of 2024, report of a use of a nitazene analog, perhaps slightly worryingly from a 16-year-old who had, at that time, quite considerable drug use experience over the years. Did have this, the compound tested, which is I don't know how common that is amongst drug users to have their drug samples tested prior to ingestion, or not. But in this case, this user certainly did, and it did match what they thought it was, and did a fairly sensible, as far as drug taking can be, increasing the dose over a series of doses, and recording the effects that they saw at particular times. So again, these sort of things give us some real world experience, if you like, of what people are experiencing when they take these compounds. So, of course, as far as opiates and opioids go, we've seen the sort of evolution from the more traditional drugs, so morphine and heroin, of course, oxycodone and the OxyContin crisis in the United States, leading on to the fentanyl and fentanyl analog crisis that still goes on now. And then more recently, of course, with the banning of some of the fentanyl analogues and the precursors used to manufacture those fentanyl analogues has given rise, in part at least, to the presence of nitazene opioids in the illicit drug supply. So nitazenes go back sort of around 2018, 2019 is where they were beginning to be seen in the United States. And then the question, I guess, is what next? Anecdotally, there seems to be some sort of a resurgence, if that's the right word, of fentanyl analog use in the United States. So fentanyl analogs are still fairly commonly seen. Fortunately, in the UK, we didn't get too many fentanyl analog cases, certainly nothing like the scale in the United States. But again, hopefully, if nothing else from this set of slides, you'll have some reassurance that the analytical testing capabilities are there in some, if not most, labs in the UK to identify these things. There's some really useful work coming out from laboratories across the world. In particular, Christoph Stove's group over at the University of Ghent have done some fabulous work looking at the relative potencies of new synthetic opioids. So they have an in vitro functional assay, which is a really useful tool, and this provides us with a number of useful things. Number one, the relative potencies of the different analogues by measuring formally the interaction with the receptors, but in an in vitro model. And then secondly, because they've got a whole host of these different analogues now, they can begin to look at which functional groups within the chemical structure of the different analogues are responsible for increases or decreases in relative potency, which gives us some idea if a new analogue comes along as to whether that analogue is likely to be more or less potent than compound X, or it's normally equated in terms of potency to either morphine or fentanyl. But by doing this predictive work and starting to look more formally at how the different parts of the chemical structure contribute or otherwise to potency, gives us a clue for new compounds that may come along in the future. So this is really invaluable work. In terms of the timelines of the nitazenes, as with the Upjohn compound that we saw on the first slide, these compounds were initially developed as pharmaceuticals. In this case, for the nitazenes, 2 nitazenes were developed in the 1950s by Sieber. There were a few and less than a handful of cases of nitazene as a substance misuse in the late 90s, but really driven by this increased legislation, as I mentioned, on the fentanyls and the precursors of fentanyl analogues. We saw the rise of these in 2018, 2019 in the United States, and the first case for isotretinoin there reported in 2019. In 2022, to potentially exacerbate the situation, there's been a comprehensive ban by the Taliban on opium production, which they estimate will cause about an 85% reduction in the availability of opium. So again, we may see that contribute to increased cases of other non-heroin related opioids. And most recently, late 23, 24, we've begun to see N-des-ethyl and methylated nitazene analogues detected in samples, and either as compounds in their own right, as well as I'll come back to, the importance of measuring the metabolites of these compounds as well. And late 2024, we had a list of nitazene opioids that in the UK were classified under the Misuse of Drugs Act, and that list is shown here with the chemical structures. So a couple of things to point out here is there are some compounds with more than one name. So the N-pyrrolidino-etonitazene also goes by the name of etonitazepine. So as with the cannabinoids, perhaps to a lesser extent than with some of the synthetic cannabinoids, knowing the chemical names and the convention of the naming of these compounds can be important. I've highlighted in red those nitazene analogues that contain a nitro group at the R1 position, and I'll come back to the importance of that when it comes to biological testing a little bit later on. But you can see of the list of nitazene analogues that are now classified in the UK, a large number of them are these nitronitazene compounds. The other thing that's important in terms of how we measure and detect these compounds is that a number of these compounds have the same chemical formula. They just have a slightly different chemical structure. So the same atoms within the structure, but just put together in a different way, which means as far as a mass spectrometer goes, they are the same mass. We have tools to help distinguish between compounds which are the same mass, which is useful. And again, I'll come back to that a little bit later on. Just in terms of analysis, this is a slide that I always put in just to show the complexity of what it is we're having to look at in biological samples when it comes to drugs and their metabolites. So if you were in a nightclub in the 90s and saw a pill like this, chances are you would expect that compound to contain MDMA or ecstasy. And just by way of example, if you take the chemical formula for ecstasy, it doesn't work so well over a webinar. Normally I would ask the audience here how many chemical compounds they think exist that have the same elemental composition as MDMA. The answer to that is almost 16,000 at the last time of checking. So there are almost 16,000 other known chemical compounds that have the same molecular weight as ecstasy. Obviously different structural formulae. Some of these compounds will be incredibly rare, but still that number is quite high. And so in the laboratory, what we need to do is to try and be as certain as we can that when we're saying a compound is ecstasy, how many different compound identifiers have we got to say that that compound is as far as we can possibly say ecstasy. So looking at the analytical tools that we employ to make those decisions. What we have in the laboratory is a chromatographic system. So we use liquid chromatography, but the gas chromatography is an alternative. A compound or two compounds with the same mass might have different chromatographic properties. So the retention time is characteristic of a compound. We measure the mass to charge ratio of an ion, which may well, as I've mentioned, have other compounds with that same mass to charge ratio, but it's an identification criteria nonetheless. We also have naturally occurring isotopes of these of the atoms within the molecular structure, and the ratio of those isotopes is characteristic. But then more importantly, and you'll have heard of the term tandem mass spectrometry, this is where we take an ion and we fragment that ion. The way that an ion fragments is characteristic of the way that the molecule is put together. So you get characteristic fragment ions, and the relative responses of the different fragment ions are characteristic under fixed conditions. So we have all of these extra criteria aside just a chemical formula to be sure that we're saying this compound is what we think it is. Coupled with the fact that we also will have circumstantial evidence and, importantly, the presence of metabolites is really important for us. And I'll come back to the metabolism of nitazenes in the next couple of slides. A take-home message for everyone is that with all of these things, really it's very important for us to have reference standards. So we will buy in a certified reference standard for metonitazene, by way of example. And that then allows the laboratory in our own setting to determine some of these identification criteria. So whilst we can get a good feel for what a retention time may be, nothing actually beats having that chemical compound in your library of reference standards and being able to run it through your own systems and your own instruments. The good news on that front is that maybe 5, 10, 15 years ago, the availability of reference standards for novel psychoactive substances was not as good as it is today, and they were horrifically expensive. That both availability and the cost of them is improving dramatically, and there are now some reference standard manufacturers who are beginning to make standards for compounds that might appear in the illicit drug market potentially before they even do. So again, trying to address this issue of staying in front of or ahead of what people potentially are using. The availability of reference standards for metabolites and breakdown products is less well developed, but again, there's good progress in that area too. One thing that's unique to the nitazene class of compounds when it comes to how they behave when analysed using mass spectrometry is that the fragmentation that I've mentioned, which is useful as a compound identification criterion, is fairly limited. So they really only fragment well at the two bonds shown with the blue lines on the structure on the left. So most nitazenes with this diethyl group, diethylamine group, will give characteristic fragments at a master charge of 100 and of 72. So you don't always get much structural information from traditional mass spectrometry fragmentation alone. So this, if nothing else, makes the role of chromatographic separation even more important. And there are alternative fragmentation models that can be used, but they require specialist instrumentation to be able to undertake, which is certainly not available widely in a number of laboratories. The other thing that's worth mentioning here, specifically with regards the analysis of these compounds by mass spectrometry, is that despite the fact that these are incredibly potent opioids, which infers the concentrations in biological samples are very, very low, they ionize incredibly well. So two compounds with different structures, weight for weight, will give different responses on an instrument. Because of the chemical structure, and in particular those ones with the nitro group, the ionization in a mass spectrometer of these compounds is really very good, which means we can, with modern instrumentation, very readily get down to very low sub-nanogram per mil concentrations, even in some screening approaches. So despite the potency of these compounds and the required sensitivity, the characteristics of the molecules and the ions that they produce is such that the sensitivity is less of a challenge than it is for some other classes of compound. So that's good news. Again, just a slide that I always include, where we're looking for these compounds in biological samples is very different to their analysis in, for example, seized drugs. We're dealing with far, far lower concentrations. We've got the added complexity of the sample matrix, so we have to do a quite extensive sample cleanup procedure. And really the main thing is the complexity of how a drug is transformed. That may even be during the dosing. So we've got examples where pyrolysis products are produced during smoking. So one thing we're keen to work on is to look at the possible pyrolysis products of some of these nitazene analogues when they're ingested via smoking. There are some compounds known to biotransform when co-ingested with other drugs. I'll give you an example of that, but that's something that we sometimes need to consider. We need to assess how stable these compounds are, and I'll give an example of that with relation to some of these nitro-containing nitazenes in a second that we've recently published, and the fact that these compounds are sometimes fairly extensively metabolized or transformed into different products. This is just to show you an example of something we did with synthetic cannabinoids a couple of years back, taking the example of cocaine, which when co-ingested with alcohol gives you the specific transformation product of coca-ethylene. We had evidence to suggest that some synthetic cannabinoids can undergo a similar transformation when co-ingested with ethanol. So we need to look at the chemical structures of the nitazenes and see whether that's a possibility for this group of compounds. But even for the nitazenes themselves, without worrying about co-ingestion for other drugs, and by virtue of knowledge of structurally similar compounds and common phase 1 human metabolic pathways, we would predict that metonitazene, which again, just by way of example, if you put that chemical formula into PubChem, there are 12,500 compounds with that same molecular formula, reinforcing the need for us to have good chromatographic separation and powerful mass spectrometry to identify the compounds. But for that compound alone, we would expect N and O desalkylation, hydroxylation at various different sites on the structure, and then reduction and acetylation of that nitro group. So it's those last 2 points that I'll come back to with a specific case example. But you can see already that from one compound being ingested, you can give rise to And each of these compounds that is formed by metabolism will, of course, be at a lower concentration than the drug itself, which are already very low. We get a very complex picture very quickly. So just to take metonitazene as an example, there's a table here, and I don't expect this to. The point of the table here is just to show how many different metabolites potentially there are that should be included. So a library full of compounds, when it comes to being able to screen for metonitazene, is not just a case of adding metonitazene itself to your library. That's good to have, but it won't always be present. And you need to think of the metabolites that may be present and add those to your library as well as the metonitazene itself. And you can see again, if you look through the precursor ion, so the fourth column in that table, there are a number within that column where two different metabolites have got the same mass to charge ratio. So again, our ability to separate and identify those independently is important, and certainly not all of these are available as reference standards. So there is some work still to do in that area. Again, just looking as an example, research going on globally is really useful for us, where in vitro models are used to identify more formally, using human liver microsomal studies, what metabolites we would expect to see. And again, this is just nice in vitro confirmation of the metabolites that we would expect to see, matching that which is in the table previously shown. But again, showing the complication and the number and the extent of metabolism of these still relatively simple molecules, and combinations of them being present in biological samples is really problematic. But there's two sides to that coin. Problematic in terms of the number that need to be covered. But if you see a drug and 3 or 4 metabolites, that's good confirmation that you're measuring the right thing as well. Some of the nitazenes share metabolites. So if you look at metonitazene and etonitazene, when you get the O-desalkylated metabolite, which the methyl and ethyl groups there become hydroxyl groups through the purple dashed lines, you get a common metabolite to both of those compounds. So in this case, as well as measuring the metabolite, any presence of the parent compound would be necessary in order to say a person has ingested either metonitazene or etonitazene, or of course a combination of the both. So one thing we've looked at recently, and we were doing some work with the and we had a case that intrigued us slightly and gave us some not cause for concern, but some reason to review the analytical data that we had both produced. And what it led us down the path of thinking was to learn from examples of nitro-containing compounds, such as shown on here, clonazepam. So you can see on the left the structure of clonazepam, which again has got a similar nitro group to a large number of these nitazene analogues. And benzodiazepines, nitrobenzodiazepines, which include clonazepam, but also nitrazepam and flunitrazepam, are known to undergo this reduction to an amino group and then an acetylation to an acetamido group. And this can happen in vivo, but it can also happen as biotransformation from microorganisms. So this is relevant to post-mortem casework, and particularly if there's a large post-mortem interval. So it's a well-known phenomenon, but it hadn't been previously reported. Particularly the acetylation stage of this reaction had not been reported for nitazenes in the past. But we had a case from back in June 2023. I say this was just published at the back end of last year in Journal of Analytical Toxicology. And the reason it was interesting to us is that the initial analysis, which was targeted, showed the presence of, albeit a very low concentration, of metonitazene. And the confirmatory analysis was commissioned, but the metonitazene was absent. Importantly, this was in an unpreserved blood sample. So here we have some mass spectrometry data. So on the top you can see we call this the total ion chromatogram. That's the chromatogram for used for a screen, if you like. And then extracted from that, you can see the expected retention time and peak that we would get, and the corresponding mass spectra shown in the bottom right for a metonitazene reference standard at a concentration there of 10 nanograms per mil. And that was absent in our patient in our case sample. Sorry. So this is just I've just showed you this table before, but what we're able to do with the instrumentation that we use is to retrospectively go back over that screening data to see whether any of these other compounds are present. And so the column on the far right there shows which compounds were present in the sample that we received for confirmatory testing. So importantly, on the top row, the metonitazene there was not detected, as I've mentioned. So it was present in screening, not detected when later analysed as a confirmatory test. What we did see was the 5-amino metonitazene, which we would expect to see because of the similarity with the nitrobenzodiazepines, where you get a 7-amino clonazepam, 5-amino metonitazene. And we also saw a response characteristic of the 5-acetamido metonitazene, which has not previously been reported. So you can see the question mark there for the retention time of the third row of that table, because we're unable to confirm that with a retention time from a reference standard, because that reference standard doesn't exist. Fortunately, not many of the other metabolites were detected, which suggests either they are present at very, very low concentrations, or they're not formed in the human metabolic pathway to concentrations which we can detect. But there were a couple of others that gave suggestions at low concentration. The important thing here being, if the method was set up to test for metonitazene alone and not to include the amino metonitazene and the 5-acetamido metonitazene, we would have missed the presence of metonitazene in this case if we only had access to a single unpreserved blood sample. And here's the data then, just to show. So this was a sample, as I say, screened positive in a preserved blood sample for metonitazene. The plain sample coming through to our laboratory shows in trace B the absence of metonitazene itself, but the potential identification in traces C and D of these two biotransformation products. This again, just to reinforce, the metonitazene calibrator shown on the left, we get a nice peak at the retention time we expect to see, just over 6.5 minutes, was absent in traces B and C. The acetamido metonitazene and the 5-amino metonitazene were absent in our reference standard, which is as we expect, but they're both present in the case blood sample shown on the right. And we have the mass spectrometry data, including the fragmentation data, to confirm that. Again, just to look at the relative retention comparison that we know for clonazepam, just to again give us some extra certainty that these putative metabolites or degradation products are what we think they are. The relative retention times of the compounds were in agreement, and full details of this are available in the publication that I just showed. So the take home there for the stability of these nitronitazenes, which I mentioned earlier, was a large number of the nitazenes that are now controlled in the UK. Wherever possible, use preserved samples, so fluoride oxalate preserved samples. If you've only got access to plain samples, then the inference is that the inclusion of metabolites and or degradation products is really important to minimize the risk of false negative results. All of this has become possible by drawing an analogy with older drugs that we know a little bit more about. The nitrobenzodiazepines in particular were useful for us here, coupled with the published in vitro metabolism studies. And inevitably, I think there will be a role in the future to think about prediction and machine-based learning for identification, not only of new compounds that may be produced, but also how they may be metabolized and the relative abundances of the different metabolites that might be present in different biological samples. Just quickly, a note on a couple of other groups of compounds that I'm sure you have heard of and where we're looking next. So xylazine again is a compound that has been portrayed in the media as another one of these zombie drugs, although a large number of classes of drugs seem to be reported as zombie drugs. Xylazine just being one of those. We've been measuring xylazine in samples now for a good couple, if not a few years. So again, good coverage of this. It's another compound that ionizes very well. So in terms of detectability, if you like, is pretty good. And we've seen this both in heroin contamination with and without the presence of nitazene opioids. Medetomidine is a potential compound that's sort of coming next, if you like. So alerts from groups in the US. This is an example of a publication that comes out from a laboratory in the US. It's the CSFRE, which is the Center for Forensic Science Research and Education. A really productive labs in terms of output, and sort of these alerts are really, really very useful, and they actually contain quite a lot of analytical information which help us ensure that our screening methods are capable of picking up these compounds, hopefully before we need to see them, rather than us having to do things in retrospect. So medetomidine and dexmedetomidine is something that was alerted back in 2023. We were able to very quickly buy a reference standard for that and make sure it could be added to our library. Xylazine, of course, now has been banned, as have these list of opioids. So again, these are the newer ones that are on our list. The one at the bottom of the group, the two compounds shown at the bottom, are interesting because there's as yet no commercially available reference standards for those, but we're in the process of asking and liaising with some of our reference standard manufacturers to ensure that we have availability of reference compounds for these analytes, so that we can detect them. So in summary, nitazene opioids, I'm sure they've been fairly well publicized in the media, not always scientifically correctly. Sometimes nitazene is sort of a catch-all term, and hopefully you'll agree that nitazene as a single compound is far from the truth. There are a number of nitazene analogues, and each of them give rise to potentially a large number of structurally related metabolites. But despite the fact that they are incredibly potent, and therefore the concentrations we're expected to be able to detect are very low, they ionize and can be analyzed by mass spectrometry very, very well. And because of our knowledge of metabolism and our ability to quickly add drugs and their metabolites to our libraries, the nitazene opioids can be relatively easily detected. And I think the good news is that awareness in toxicology labs in the UK and globally of these compounds is very good. And if not directly covered in a laboratory, there are case circumstances where people would be able to perhaps suspect a nitazene opioid might be present and do some additional testing, or refer samples to a laboratory that can do the testing. So I think in comparison to where we were just a few years ago, the risk of missing the presence of these nitazene opioids is reduced. I don't think it's entirely gone away, but it certainly is reduced. The modern instruments that we use, as I say, allow us to add new compounds relatively simply to our libraries. That doesn't mean that as soon as a compound's added to the library, the methodology is ready to go. There are always complications. The stability of these nitro-containing nitazenes is just one example where, just adding a compound to a library doesn't mean that you're immediately able to detect it. You need to understand the toxicology, how the compound potentially will change once it's ingested, and depending on which route of ingestion. So all of that, I think, underpin and reinforce the fact that sharing of analytical data, reporting of cases, case reports is really very important for us and for all labs to keep as far as we can up to date with the drugs that we're looking to see. And of course, the nitazene opioids and synthetic opioids more generally are just one class of compound. We're doing similar things with novel benzodiazepines. We've got synthetic cannabinoids as a class of compounds. There's still other sort of stimulant type novel psychoactives that are finding their way into the illicit market, and of course an ever-growing number of prescription drugs that we're looking to be able to measure. as an example there, and whether it will stimulate any discussion or questions. We're also working on methodologies to be able to detect, for example, the GLP-1 agonists, the semaglutides, and the Ozempics, and the like. So, our job as a busy forensic toxicology lab is never done. Every case that comes through is a potential new challenge, and hopefully I've just reassured you, if nothing else, that we're doing our very best to be able to ensure that our scope of coverage remains as up to date as it possibly can be, to risk missing the presence of compounds that may be of relevance and importance. And with that, I think I'm just about on time and have left time for questions. Thank you very much.
Well, thank you very much, Lewis. I think there should be some questions now. I'm just waiting to see what comes in. At least there's one person asking a question. Angela Burns asks, is there any comment on urine stability?
Yeah, so we were fortunate to have an MSc student who did some very preliminary stability work, and we need to do a little bit more work to get that in a form that's publishable. But what we found for a selection of 9 nitazenes is that in preserved urine samples, they were pretty stable over time, and that included storage in the fridge, storage at room temperature, and storage through freeze thaw cycles. Again, the other aspect to that question would be, in the urine, as well as the stability of the compounds themselves, what is the relative proportion of the metabolites or breakdown products, and then how stable are those? So there's still some work to do, particularly with regards to the stability of those latter compounds, which to my knowledge hasn't yet been done. But the compounds that we tested looked pretty stable under a range of conditions in preserved urine.
Perhaps you should say what you mean by preserved sample in terms of urine. It's not just a matter of putting it in the fridge, although that helps.
No, it's preserved with fluoride oxalate, so sodium fluoride and potassium oxalate in the sample. So we send those, we send sample collection tubes for our post-mortem work to the mortuaries. So tubes with fluoride oxalate preservative pre-added for a 5 ml volume of sample, and we ask for those for blood and for urine, and also for vitreous humour samples to be collected into those tubes. We ask for the collection of plain samples as well, but our preference when it comes to quantitative analysis for most compounds these days is to do so in the preserved sample. And the metonitazene is a good example where measurement in the plain blood would have potentially risked us missing the presence of the metonitazene itself.
I don't know, I can't remember whether we've had these or not, but the next question is, can they be detected in other tissues if the body is decomposed?
That's a good question. I'm afraid, yeah, we have not had any cases where we've only had a liver sample, for example, or a muscle tissue sample, and have detected a nitazene in it. Our screening method for tissue samples is screened against the same library as our blood and urine samples. So if they were present, we should detect them. There may be some slight differences in how low we are able to detect because of the complexity of a liver sample versus or other tissue sample versus a blood sample. But we've had no cases of a nitazene detected in a tissue sample. The ones we've had have all, fortunately, we've been able to access blood and or urine. So I don't know of any liver or tissue cases with nitazenes detected.
Okay, you've presented the details of the analysis of these, and obviously we're a full function laboratory with a room full of mass spectrometers of both high resolution and low resolution mass spectrometers. We need that to identify some of these compounds. Is there an issue with the loop, perhaps, which is turns up at festivals and tests people's drugs? But is there an issue about them lulling people into a false sense of security because they can't detect some of these compounds and saying, well, there's nothing in there, or it's some other compound if it's a mixture? And then people go ahead and take them and find out, get a bit of a shock.
Yeah, potentially there is. So what you can do in a field, I mean, and literally in a field, um, is certainly limited, and I think they acknowledge that there is limitations to the methodology used. One of them is sensitivity, particularly when you've got low level contaminants, if you want to call them contaminants. So the presence of a nitazene in a tablet, for example, at low concentration, potentially would be missed using the techniques that are used. There are advances in field analysis, so you can now purchase portable chromatographic systems, portable mass spectrometry systems that offer some promise for being able to detect these things at lower concentrations and presence in mixtures. But they're still very much in their infancy with regards testing specifically for drugs and things like synthetic opioids, because they need to be added to libraries and the like. So whilst they're available, they're not widely used. So the limitations of in-field testing when you're looking at, for example, um, spectrophotometric methods, so FTIR and Raman spectrometry, that there is a risk of missing the presence of low-level nitazene opioids in, for example, 2CB pills, which as per Wedinos has been detected in the past.
Right, we've got a couple more questions in the Q&A. Under the Misuse of Drugs Act, is it only possible to ban specific compounds, or can a chemical class be classified? Lewis?
Oh, that's I'm not sure.
I'll take this if you like.
Yes, yeah.
And essentially, I mean, this whole business of banning things, I think it makes the government feel good. So you saw on one of Lewis's slides that the Right Honourable Dame Diana Johnson banned xylazine. Well, that's great. It made her feel good, I'm sure. Did it make any difference to users? I doubt it very much if it did. When you buy these things, if you buy these things, you don't ask the supplier, is this a banned compound I'm buying? Because you have no idea whether it is or not. So banning them is really, well, we ban murder, but it still goes on. And I think drugs will still be sold. And there is some evidence out there that the prohibition of drugs actually has some harmful effects in its own right. So we need to be thinking about this as to what our policy is. Yeah. If the government really wants to do something, it should be funding. It should be funding a little bit more people like Tic Tac and Wedinos to pick these things up, to look at them and actually get it out there. But you just ban something, and the chemical roulette happens, and we get a different compound found, which gives the labs another headache to find what that is.
Yeah, and the interesting one was the one on the last slide there, where we've got a compound that's banned that we can't buy a standard for.
Yeah, I mean, it really is the unknown unknowns that we have a problems with. I know Donald Rumsfeld got a lot of stick for that quote, but that is the issue. The unknown unknowns are what are the problems, because we won't know it until it appears. Do you think have we had any CSF samples?
No, I can't remember the last CSF sample we got at all, let alone one containing a nitazene. So
we don't know if it's stable in CSF,
no.
but I would imagine it should be. It's mostly it's a low protein containing fluid, and if you have a sodium fluoride, should be reasonably stable.
Yeah.
All right. As this is a fast-moving area, what changes? Which changes all the time. What is your approach, even with reference standard availability, validation, and verification for accreditation purposes?
That's a really good question. Certainly for us, so our accreditation is to ISO 17025, and it's for a very prescribed list of compounds. So for us to develop, verify, validate a compound takes time, takes resource, and as you say, it's fast moving. So it may well, by the time if we were to do a formal accreditation under 17025 for a named compound in a named matrix at a named calibration range, would possibly take as long, if not longer, than the duration of the lifetime of that compound within the marketplace, if you like. So it's moved on to the next one. So we validate methods. We have a sort of standard approach to method validation, so we can get things like limits of detection, limits of quantification. We still run quantitative assays as per a standard operating procedure with independent calibrators and quality controls at multiple levels, etc. So we do that for every compound, but it's not formally validated and listed on our 17025 scope of accreditation. So you've got a validated and verified method. It doesn't have the ISO stamp on it, but all of the work, of course, is undertaken in a lab that does have a quality management system that ticks the boxes for ISO 17025. But it's a really good question. It's a really good question, and the idea of a flexible scope of accreditation is something that's talked about, and I don't know how easy it is to achieve and enforce.
Yeah, I think the United Kingdom Accreditation Service have got just can't get their head around it. The Americans, in their equivalent ANAB, they accredit the laboratory for toxicology analysis. They don't accredit individual compound analysis. It's the laboratory's accredited for its approach, not for actual compounds, named compounds. Difference of approach, but UCAS don't seem to be able to get that through their heads. We don't know what's coming in the door from one day to the next. There's a question here about, is there any data on oral fluids? And I don't think there is, but Lewis, maybe I've missed it.
No, not that I know of. No, my guess is if there were any, it would come from confirmatory testing in Europe, where they're doing some related to drug drivers and things. But I don't know of any data specifically to the nitazenes in oral fluid. No.
The trouble with oral fluid is you need either a live patient who's conscious to get oral fluid, and quite often what we're seeing are coming from post-mortem cases where obviously we're not going to get oral fluid, and perhaps unconscious patients, again, it's difficult to get oral fluid from, so we don't see much of that.
Mm-hmm.
Right, are there any more questions, or are we about to close the Q&A session? One last chance, perhaps, to ask a question. Okay, well, if that's it, thank you very much for listening to Lewis and coming along to the webinar. We haven't got another one planned from me, but I believe there are more. The SAC in toxicology has got some more webinars planned, and watch out, and we'll publish that as they come up. So thank you very much for your attention, and thank you very much, Lewis, for that presentation.
My pleasure. Thank you, everyone.
Bye.